Okay, so welcome everyone, and thanks a lot to be here. First of all, thanks for the invitation, Christian, for to give this talk. That will be a very fast talk about my long-term research. So, as you can understand from my age, it has been long, several decades. So I will just give a brief presentation of several of my long story. So I'm Margarida Matos, as I was saying, and I'm from the Faculty of Sciences at the University of Lisbon, and a researcher at the Center for Ecology, Evolution, and Environmental Changes. In my talk, as you can see the title, From Nature to the Lab, the role of history, chance, and selection during adaptation to novel environments. So basically, what do we do? So in our longer term paradigm has been to study, as Chris was saying, a real-time evolution of populations, in my case, Drosophila subobscura, since the moment they entered the lab from collections in the wild. And this has been to basically to understand the adaptation to a new and confined environment, so contrasting environment but in control, but anyway, a new environment, so the dynamics of it, also characterizing the evolution changes of these small and confined populations in several markers. Remind that Drosophila subobscura, particularly at the start, and still is not really a model organism, so we use neutral markers like microsatellites in chromosomal arrangements, and then we more recently started using evolving reseq analysis during lab adaptation. And even more recently, and that will be the second part of my talk, we have been addressing thermal adaptation after lab adaptation. But the main talk will be about the lab adaptation. Just to recall, all of you know, but anyway, what is experimental evolution in a very fast way? It is the study of the real-time evolution of populations under control conditions. And so basically, I like to say that it's evolution biology in its most empirical sense. And it has several conditions. It allows to analyze evolution potential population, but we have to be careful about several issues. One essential issue is the number of founders. So except when we want to characterize the effect of a few founders, a high number of founders is an important issue if you want to address dynamics of outbred populations. A big population size throughout generations is obviously a corollary of this to maintain diversity as much as possible. And finally, replication. The level of replication in experimental evolution is the population, because it's a unit of evolution, as you all know. So this is also a very important issue. And I have here some a review in 2012 that you all know probably, and a book where I have a chapter that may be interesting also. So why study real-time lab adaptation? First of all, because the dynamics of this adaptation is another way of studying local adaptation in general. And this is my main reason. But anyway, also, and that was the start of my PhD, it was because there were controversies about the limitations of using populations lab adapted for many generations or for a shorter number of generations. And so the analyzing how they change is also an interesting issue in itself. And finally, because it can, help characterize how much ex situ conservation problems can lead when the populations are reintroduced, and this is a way that you can ask for financial support, but it's not really my main interest. It's more about local adaptation. And so that there are no confusions, you can see here one way of addressing my questions. How much the populations change throughout generations? And you see here the evolution trajectory of one important fitness-related trait, and this is the goal of my studies, lab adaptation, that I also call domestication. And because of this, some time ago, I was interviewed by some journalists that thought that I was training Drosophila. And this was a laugh, because after, this was many years ago, but they thought that I was training Drosophila. That is not what I did. Okay, so what are my main questions? So first of all, how fast and what is the pattern of adaptation to this novel environment? and how much do different traits differ in this pattern. So predictably, those more related to fitness will have a higher dynamics. Also, does this adaptation lead to convergent evolution, and how much the role of history, chance, and selection occur during this process? Connected with this, how repeatable it is, this evolution dynamics among populations, and so in a sense, how predictable the pattern is. And finally, do different biological levels tell similar or different stories? And so all this have been throughout my, all these years have been addressed by my research, or by the research of my team. So our experimental design is very simple. So we start by collecting flies in nature, so to found laboratory populations. Then we form replicate populations, and we use as controls populations that were already in lab for many generations, and so assumed to be in equilibrium. So this is a way of analyzing this when we ask for lab adaptation processes. We have to maintain the flies, the populations in a very tight conditions, since we are testing whether they converge to the same environment throughout time. And because of this, then we can collect data by periodical assays to trace evolution trajectories of life history traits, and study also the genetic level, collecting samples throughout these generations. And then we can found new populations and repeat it all over again as a function of new questions that arise. The maintenance of our populations is the standard way of maintaining populations, as in Melanogaster. The difference is that subobscura is maintained in general at lower temperatures. And so generation time is like the double of normal generation time of Melanogaster, 28 days. And we maintain in discrete generations, which is also convenient for experimental evolution, meaning non-overlapping generations. So meaning that the mothers and daughters do not coexist at the adult stage. So when we collect the eggs for the next generation, we discard the previous generation, the adults, or we can freeze them for later genetic analysis. And so now, very fast, I will speak to you about a little bit about our studies across several years, starting from collections in Portugal. So this is like a phylogeny of our laboratory populations of Portugal that started, as you can see here in this axis, several years of foundations, and we will shorten this talk to starting in 2001. Okay, so we already had studies before, but in 2001, because before we were studying only populations founded from Sintra, we decided to change a bit and collect not only from Sintra, but also from Arrábida. So Arrábida is a location that is close to Sintra, but 50 kilometers across the Tagus River. And so we collected flies in Arrábida that we call AR, and again in Sintra, as before, that we call TW, and used as the NBs founded in 1990 as our control populations. And so here you can see the graphs, the figures representing the evolution trajectories of the TW on the left side and the VAR on the right side of the evolution trajectories for age of, early fecundity, okay? And always as a different to the controls, okay? So the NBs as controls. So you can see that the values are negative, okay? Because, and this is generation, okay, in the x-axis. And so as generations proceed, you see that they are getting closer to the controls. So they are converging to the control levels, both in TW and in AR. And the parallel of these regression lines indicates consistency between replicates. And so we found general adaptation across all populations, but with differences because the TWs were faster than the ARs. And so we had this important question. Are these differences due to the natural location of the populations or to sampling effects? And this because we had just a sample collection from each location, and we just replicated our populations already in the lab. And so the differences might be due to sampling effects. And so we had the idea of going back to these two places, to Arrábida and Sintra in 2005, And this time in each one of these locations, we collected 2 samples. We got 2 samples separated from few days. And so we called one sample A and another sample B. And then, as usual, by generation 2, we divided in 3 replicates, each one of them. And so, as you can see, this is the previous foundation, AI and TW, and in 2005, as you can see, we got 4 different foundations, 2 from Margarida and 2 from Sintra. And the rationale was, if the differences that we observed are due to the local, to the geographical location, then the differences between the 2 sets from Sintra will be bigger compared to these 2 sets of Margarida than in each one of the locations between A and B. And so this was like a hierarchical design. And we then decided also to analyze this in comparison to other studies that we already have, the AR and TW, and also because we had also data from 1998. And so we joined all these analyses in a meta-analysis to ask whether, what is the importance? Well, the importance of the time, of the year, of collection, of the place, Margarida or Sintra, and also of sampling effects. And so this is what is depicted here very briefly. Can you see, if you can see here, you see 3 graphs representing early fecundity, starvation resistance, and peak fecundity. And for instance, looking at early fecundity, here is a very brief summary of the evolution rate, okay, of the first 20 generations. So this is the slope of this of the regression lines that you saw earlier. And you can see here that, for instance, NW was the faster to respond to selection in early fecundity, and the others were slower. And the error bar means the differences between replicates. And so we asked the question whether the slope is significantly different in different, dimensions. And so we tested for the adaptive rate differences, and we saw that the differences were more due to the time of collection, that is the year of foundation, and for differences between locations for peak fecundity, so it changed between traits, between Sintra and Margarida, and also some sampling effects, but not much. We found that in 2005 only for starvation resistance in Margarida. We also have long-term studies in these populations, and just for you to see one nice figure about this, for more than 100 generations, we found that the populations tend to do similar patterns, getting closer to the control. So we never got like a superfly getting better than the controls. So the general conclusions at this part of the study, we found clear adaptation across populations. In spite of general convergent evolution, we found some differences between them, as might be expected. And I didn't tell this story in detail, but later we analyzed microsatellites of our founder females and then of the very close generations, and we understood that in fact the founders were very similar. And so all the differences that we saw before between Margarida and Sintra, for instance, were really due to the 2 events after foundation in the lab. It might be expected because, Portugal is a small country, and so in fact we had a very big population, one single population. Anyway, recent long-term analyses have shown that the long-term patterns are useful because not all the time they are predictable. So having more data is more important. Okay. So what are our present populations in questions? So we were a bit sad because we wanted to test whether different genetic background from the origin affected our dynamics. And so we understood that we didn't collect the flies where we should. And so the question remains, what happens when populations with a deep historical signature adapt to the same environment? And you recognize this, from a textbook idea that you may have an expectation of evolution towards a single adaptive peak, like in this graph above, more like the Fisherian approach, or you have, a rugged landscape where, depending on the genetic background, populations can get stuck at different peaks. And so having a deep historical signature would be the idea to test this in the lab. So the question is it possible for populations to converge under the same environment? And again, how much do different biological levels tell the same story? And so, taking advantage of the fact that Drosophila subobscura is a palearctic species widespread in Europe and has also a very clear climate variation of chromosomal inversion polymorphism in body size, and this is repeatable in more recent foundations of the South and North America, so indicating a signature of selection. We ask the question, what happens when populations from contrasting European latitudes are brought to the lab and put in the new environment? And so we, in 2010, again, made collection, but this time going further. Okay, we went to Groningen in the Netherlands, to Montpellier in the south of France, and again to Adraga, and collected flies to found new populations. That then we called 8AD for Sintra, Gro for Groningen, and EMO for Montpellier, using as controls our TW that was founded in 2001. And so the first thing was, are there differences at the start? Because if they are not, we cannot ask the question whether they will converge. And so in a very early generation, we tested this and we saw differences, clear differences between them. For instance, in early fecundity, you can see here, and this time at the x-axis is only the different foundations, AD, MO, GRO, and TA, and you can see that they are very separated. This is the control, and this is the Groningen populations, Montpellier, which was the lousiest, and AD. And all of them show the same thing. So high initial differentiation between them. So effect of prior evolutionary history, check. So we can now test whether they will converge. And so then we collected data throughout several generations. And this is what we got. So you can see here, again, for early fecundity, for instance, this right graph, you can see now, for simplicity, just the average slope, the average regression line of each foundation, and you can see that they start very differentiated, as we showed in the previous slide, and they get close together. By generation 15, you can no longer distinguish them. So a quick evolution convergence at this level. Except for body size, which was not very, was a little bit misbehaved. Okay, then, as I told you, there's a high inversion polymorphism in Drosophila subobscura, something that Christian would never approve to study this species. And so you can find here the 15 chromosomal inversion that had more consistent frequencies in all our populations. And we asked the question whether they will change a frequency through time. So this is the data between generation 2 and 40. And you can see that, so this has lots of information, but the first set of bands that you see here are from Adraga, and the second from Montpellier, and the third from Groningen. And you can see that in all of them, for instance, A2, they increase the frequency throughout generations. Whether in O3+4, for instance, you can see an increase, a steady increase in Adraga and no change in the others, or even a decrease in U1+2, decrease in Adraga and increase in Groningen, okay? So besides a high initial differentiation and changes across generations, consistent, but without a clear convergence. Another way of showing the difference between the life history traits and the inversions is like in a principal component analysis. On the left graphs, you can see the convergence between populations in a multivariate scenario for life history traits, whether on the right side, you can see for the inversions, Adraga goes this direction and grow on this. So convergence at phenotypic level, but only partial at the karyotypic level. Then we were able to do some evolving resequence analysis, and we focused on AD and GRO. And so we did whole genome sequencing by pulsec for generations 1, 6, 25, and 50. With the question of characterizing the initial genome-wide variation and the evolution dynamics at genome-wide level, and trying to find signs of selection in SNPs. So to test whether populations of contrasting genes converge at this level, at genome-wide level. Just to recall, but you know this very well, evolving resec is a new already with many years of experimental evolution, and so it's analysis of genome-wide temporal changes during experimental evolution. And there are progresses as we speak. So I envy you because you are in the right time for this analysis. So you can see here a review that you all know very well, probably. And this that I'm going to speak to you was published in MBE in 2018. And Sofia Seabra was the postdoc in my lab that helped analyze this data. So this is not necessary now. PulseSec is a very important tool. We measure many individuals, but does not allow to have the individual data. Okay. Anyway, so the genome-wide variation that we analyzed, first of all, we had a very, very high challenge. We didn't have a reference genome in Drosophila subobscura. So Sofia Seabra was able to do a draft genome. And so with many limitations, that's what we were able to do, and she did a magnificent work with it. And so we found more than 3 million SNPs in our data. And the first thing was, what is this dynamics between these AD and raw populations throughout time, okay, in these 15 generations. And so what you see here is a principal coordinated analysis of the FST matrix. And you see that the 3 replicas of a drug are evolving in this direction and the Groningen in this direction. So they are not converging at all. They are diverging, and this is the controls. Then we wanted to test for signs of selection. And so we were very conservative in this case. So first of all, we defined the by CMH test that you all know. So the consistency changes the frequency of the several replicates would give a sign of selection. And so the higher significance top 4,000 SNPs were used in the next step, Posiandrage and Groningen. Then we demanded that the minor allele frequency increase in the 3 replicates, with this rationale that it was more likely that by drift it would be decreased. And finally, we demanded that simulations at neutral model would show that it was unlikely to happen in this population. And so at the end, we were with a little bit more than 240 SNPs in AD and 165 in GRO. And as you can see in this graph here, we just plotting the CMH of the GRO and the AD, you can see that the values do not coincide at all. So they are very separated. And so no common SNPs between AD and GRO, which was a bit of a surprise given that they converge so fast in life history traits. And so this is again the plot, but now just for these 400 SNPs that gave sign at least in one of them. And you see that this plot of the coordinated analysis again show that they are not converging at all. So no common SNPs. Can the SNP, the population diverge across generations, okay? Another thing that is very interesting to see is the evolution trajectory of these SNPs, because we have data of several generations. And you all know the classic work by Orozco Terwinkel, by Christian Schrotter, 2012, where they analyzing also the evolution dynamics of adaptive lab adaptation in terms of melanogaster, and they found like 2 families of SNPs, let's say. So those that give a sign of selection earlier, they tend to plateau, which is this graph here. Whether when they give the sign of selection at a later, with more generations, they tend to have like a linear trend. And so this may relate with the dynamics of trade-offs, for instance, and the value of those the faster at the end at a lower frequency. So all this is very interesting. So we wanted to check whether we had something like this, but basically we didn't see this. Okay, so let me see if I can explain you. This is a very much information. So the x-axis is the change of frequency of allelic frequency change between generation 6 and 25, and the y-axis between generation 25 and 50. And this with the color is the SNPs that gave signs of selection in AD in the first period or after using all the 50 generations. And we expected to have some differences like in Christian Schrotter, but basically what we found is that we had all types of dynamics, from constant temporal changes to plateauing or even inverted direction of change. For instance, in the case of the earlier SNPs that might even go back after decreasing frequency after increasing. So basically the only general pattern that we found was a negative correlation between the changes at the two periods, which is expected because if they change a lot at the first, cannot change so much at the second. So also we found few of the candidate SNPs reach fixation after 50 generations, less than 5%, which is also in common with many studies. And these studies highlight the importance of analyzing these evolution trajectories because they are really interesting to analyze, though complicated to explain. So again, we asked the question, how repeatable is all this? Because we again had only one collection from each location. So in 2013, we again went to a drug into Groningen, and this time, so we collected a sample, a population that was then replicated in the lab, a drug population we called PT, and the Groningen we called NL. And we asked the question, are they going to do the same thing? How repeatable is this pattern? How predictable at the end? And so at the phenotypic level in life history traits, the repeatability was amazing. So you can see here now the plot of early fecundity for the four foundations, AD, PT, GRO, and NL, and they are very, very similar. Not so similar in age of first production because at the start they were different, but they converge also, so they are also predictable. Again, body size was misbehaved. Okay, and what about karyotypic level? So we also analyzed this, but this time 25 generations, to ask the question, we did not see convergence in 2010 populations. What about the 2013? And this time, we saw that it was not repeatable. Okay, so the 2013 foundations were more predictable in this sense. So you can see, for instance, O3+4, remember that it was increasing in AD and it didn't change in GRO, but this time it increased steadily in both PT and NL. And U1+2, the same. Okay, so we saw that this absence of convergence was specific of the years that we collected. We are now analyzing by genome-wide data that also indicates partial genomic convergence that may relate with the dynamics of inversions. And I think that Christian is going to love to hear this. Of course, that is connected. And at this time now we have a reference genome where we can align our SNPs and try to relate with our inversions. So the conclusions. Can historical differentiation prevent convergence during adaptation to a common environment? The answer is no. We saw a general increase in fitness-related traits, and so it suggests a single adaptive peak. But are the relative roles of history and selection similar at these different biological levels? The answer is again no. So we saw that selection quickly erased these signs of historical differences in life history traits, but they persisted at karyotypic level, though without a clear repeatability between years. And also no convergence at the genome-wide level. So basically this leads to the expectation that we have about the consistency of signs are higher, the higher level that we analyze. And now we have a new paradigm that I was telling you, that I'm going to tell you the second part of our story, which is moving from convergent to divergent evolution. So it was time to move a little bit from what we were analyzing. And so considering, as you all know, the concerns that we have of global warming, so we know that at several levels, some conservation to economical, sociological, you name it, because you know that there is a 1 degree centigrade rise above pre-industrial period and around 0.2 degrees centigrade rise per decade. And so this has been a concern, and so we wanted also to try to contribute a little bit to understand whether populations are able to respond to this. And you probably know the concept of evolutionary rescue. So when populations experience severe stress, they may get in trouble unless they are able to adapt to the new stress. And this is what is evolutionary rescue. It's when adaptive evolution change restores the growth of the populations to the values before the stress was imposed. This depends on genetic variability of population and the rate of their environmental changes. And so many researchers have been dedicated to understand thermal adaptation in populations of several levels, using several approaches. Comparative studies have indicated that there are high constraints in upper thermal limits of populations. Many studies of thermal experiment evolution have been focusing also on the capacity for population to respond to the stress in different designs, which makes the responses complex in generalizations. They are context specific. There are differences between studies, you name it. And few studies address evolution under dynamic thermal environments, meaning increases of temperature, of amplitude, etc. So you can see here a review that we published last year about experimental evolution in these setups of thermal adaptation using ohmic tools that you may be interested in. And so what are our new questions? So how much can populations adapt to climate change? So this is a question that many try to answer, and this is just one small contribution to that. What is the impact of contrasting ancestral history upon the adaptation to these thermal environments? This is not so usually done, and I think it's a very important approach. And we also analyze the genomic and transcriptomic basis of this thermal adaptation as also complement. And this is what we have been addressing in this project, Adapt Clean Warm, led by Pedro Simões. I'm very proud to say that he was my former PhD student, and now he's my colleague and he's the PI of this project. And so this is the team that is developing this, that has developed this project in the last years. So myself and Pedro Simões, my PhD student Marta Antunes, that she's addressing and analyzing transcriptomic data with much detail, and technicians and several collaborators. And so what are our new populations? You may recall them by looking at this. It's the PT and the NEL. And this PT and NEL, so the Portuguese and the Netherlands, and from now on you'll see sometimes the nomenclature changes because some papers demand that we ask northern and southern and others low latitude and high latitude, but bear with me. And so, but basically in 2019, we imposed a new regime, which we called global warming. And so the WPT is derived from the PT, each replicate paired with another replicate, having the PT as the controls, and the same for the NEL. And so basically you see here now the daily hour in the x-axis, because we have imposed also a fluctuating, a change of temperature during the day. So basically the control is the constant 18 degree, as you can see here, it's the control, and the warming started having 18 degree also on average, but the higher temperature, the hotter hour of the day started by having 21 degrees, and the colder hour of the night 15 degrees. And then every generation we increase a little bit both the higher temperature, and we decrease a little bit the lower temperature, so colder nights and hotter days, so that the average increased by 0.2 degrees centigrade, I think. And we had to stop this at generation 20, otherwise they would go extinct. Okay, so the data that I'm going to present to you, most of it, it's when by generation 23, when the hotter hour of the day was 29.4. So first of all, let me just tell you very briefly the data that we got about the phenotypic higher level phenotypes, which was basically focusing reproductive performance. So we asked the question whether populations are adapting to their new challenge. And so we tested this concept of local adaptation in our warming in control populations. What is this local adaptation? You probably know. The idea is very simple, that we expect a better performance in the environment where populations are evolving. So for instance, here, if population represented by a square adapts to environment 1 and the circle on environment 2, each one of them has a higher fitness in their own environment. And this means that the norms of reaction cross, and this is local adaptation. A more complex concept, but very interesting, is cost of adaptation. And this, because looking at these graphs, you cannot say unless you know the ancestral state of the populations. So it means that, for instance, if population 1, this population adapted to environment 1, now adapts to environment 2, it will decrease fitness on its previous environment. And so this is a cost of adaptation. We had this idea, we had this data because we know the ancestral state, okay? And so we are testing also for the cost of adaptation. And so we presented the data in that scientific reports of generation 22, which is on the left graphs, and generation 39 on the right graphs. And you see on the upper graphs, the Portuguese population that here are called southern, and in the lower graphs, the Netherlands population called here northern, okay? And so you see in each one of these graphs, you see the PT is the black, and the WPT is the red, and the equivalent for the Netherlands. And basically, at generation 22, we didn't see any significant changes between them. So the performance of the controls and the warming populations were not significantly different. But at generation 39, we detected a significant change in improvement of the WPT relative to PT in the W environment. So this is the temperature of the essence. Sorry, I didn't explain. And so this basically indicates that the WPT is performing better in its own environment, and the PT has a lower performance. What about the performance in the control temperature? That means at 18. There we found a small but significant sign that the WPT performed worse. And that's what I was saying about the cost of adaptation, because we know that their state, the previous state was the PT, and so it decreased significantly, so some costs. And then we analyzed the transcriptomic data. So we analyzed by pulse-seq the RNA-seq our populations at generation 23. So to see if the gene expression evolved, and also to see plasticity in our populations. And so basically we sampled, we pulled around 45 females around the age of reproduction, that would be the more relevant for fitness of our populations. We characterized the gene expression. And this was in a very fully orthogonal design. So we had 3 factors involved. So we had the selection regimes, because we wanted to see the differences between the control populations and the warming populations, evolving populations. We had 2 environments, because they were developed either in the control conditions or in warming conditions, after being in one generation of common garden. And so these 2 environments allowed in each one of these populations to test for plasticity, okay? And so we also had the interaction between all these factors. So as you see here, 3-factor design. And so we allowed also to test for evolution of plasticity, and also evolution as a function of history. So all these factors together. I don't know if I explained correctly, because this is a very complete design. Okay, so the first thing was applying the full model. We asked for each gene, okay, for each gene that we characterized, we asked the question whether this gene was significantly different between the 2 histories, between the 2 environments, between the 2 selective regimes. And so we applied with a 1% false discovery rate. So we defined those that were above this line, the p-value, okay? So, and we found that the most genes were significant between environments, which is plasticity. So you see here, 3,700. And so this was the factor that was more involved. So the most important thing is the expression changed with temperature. That was the normal finding. So we also found many genes involving his history, which is interesting, because our population were already in labs, for many generations, as you recall. And we found a smaller number, 700, indicating selection. So differences between the controls in the warming population, independent of environment and independent of history, and interaction between all of them. But this is such a high number of different variables. So we wanted to, reduce this variation. And so basically we concentrated on the warming environment, which is the one that is the challenge, okay? And so we defined a simpler model, where we defined the gene expression differences as a function of selection in each population, different history separated. So for the Portuguese population and for the Netherlands populations, okay? And we found many more genes indicating selection in the northern populations. So almost 4,000, whether in the low latitude more than 4,000, whether in the low latitude populations or Portuguese less than 1,000. So, and between them, 221 were in common. One important thing is that they were widespread throughout the genome. Okay, so no tendency for clustering, at least not very clear. And so this is important since they have inversion, though at this time the number of inversions was smaller. So we also made enrichment for finding molecular functions, and we found some interesting patterns. Basically, the tendency for the downregulation of binding functions, both in Portuguese population and in the Netherlands population. The upregulation the main functions were different, were more enzymatic activities for the Portuguese and more structural variants for the Netherlands. And finally, we wanted to test. This idea is a little bit crazy, okay? Let me try to explain it. So our idea was that it would be interesting to test whether, the idea that if a population has already an adaptive plastic response, it will tend to buffer the response at the genetic level. And so we wanted to relate this initial adaptive plasticity with evolution. But how can we define what is adaptive plasticity? And so we defined it this way. So we defined as adaptive initial plasticity when the differential gene expression between environments in the control population was in the same direction as the change of expression between evolving populations in their controls in the warming environment. I hope that I make sense and I explain. And so we needed to look at the genes that were plastic in the ancestral population and that responded to selection. And we looked at that both in the Portuguese population that you find in the left Venn diagram and in the northern population in the right diagram. And so we found 321 genes that were, candidates for this analysis because they were plastic and they evolved in the low latitude population and 723 in the high latitude. And so we asked the question, are they plastic, adaptive plastic, which means, as you can find here in this box, if the evolution is upregulation, then ancestral plasticity has to be also upregulation to be adaptive, let's say, and the opposite direction in the same direction in the other way. And for the non-adaptive plasticity, it's the opposite. So if the evolution leads to a high upregulation and the plasticity is downregulation, then we call this non-adaptive plasticity. And so we asked the question, how these categories are sorted in these genes. And we found that 256 genes of these 321 in the low latitude population, I mean Portuguese populations, like 80% were adaptive, had already adaptive plasticity. Whether in the high latitude population, which means the Netherlands, it was the opposite. It was most genes that responded to selection were in the box of non-adaptive plasticity. And this, in a very speculative way, it at least fits what we found, which is the high latitude populations had many more genes in general being selected or giving signs of selection, which is different. And so, in a sense, this corroborates our expectation that adaptive plasticity can reduce the response later. So basically, main findings of this project, we found that term adaptation is slow, and that is not new, many find that. We found some costs of adaptation, so that we observed, just in one case, but it was there, and it's interesting per se. We found high gene expression variation that preceded variation at higher order traits. So don't forget that by generation 22, we didn't see signs of changes in reproductive success, but we found many genes responding to selection. So gene expression and evolution seems to be shaped by an interplay between plasticity and selection. And I find this very interesting per se, though it was a very indirect way. So we have to be careful about this. And found an impact of historical background across all levels analyzed, life history traits and transcriptomic level, as we had found before in our study of lab adaptation. So the next steps we are working now is the temporal dynamics of this gene expression change. We are comparing this generation 23 with the previous generation and finding very interesting patterns. We are validating candidate genes, which has been a very hard task for our PhD student, but she's doing a fine job, and crossing genomics and transcriptomic data. That is going to be the next step. And that's also, I want to thank all my team throughout the years. So this main starting as PhD students, and now I'm very proud to see having their own careers. My PhD student, Sofia Chiabra, that did a great job in analyzing, starting as in the bioinformatics part, and several collaborators, and also many master students and bachelor students throughout the years. And finally, the financial support of Foundation for Science and Technology, my research center, and my faculty. And to all of you, thanks for listening, and I'm open for questions if you have any. Thanks a lot.